Hi guys, welcome to Membrane 6. We're going to be talking about membrane proteins. So if water-soluble molecules are crossing the membrane with the aid of a protein, when they can't cross the actual lipid bilayer on its own and it needs a protein, what do those proteins look like? So there are two kinds. The first kind are channel proteins. They can either have a gate or be non-gated, and I'll show you through models what that looks like. So number one could be a channel, or it could be a carrier, and these differ. They're still both proteins. Both are going to allow molecules to come through from the outside in or from in out, but they will look slightly differently and they behave differently. So these carrier proteins can be active or passive, and we'll look at these. On the left, the pink one there is a protein channel. On the right is a carrier. So take a look and see what looks immediately different. The channel seems to be a hose that allows something, the little red balls, to go from out to in without anything stopping it. The carrier protein, however, never has an opening that's both to the outside and to the inside at the same time. So it's more like a hinge mechanism, where if you're opened to the inside, it'll look like this, and if this can tweak and open in the opposite direction and let things out. Okay, so a carrier never has two openings. A channel does. What do they have that's similar? Both the channels and the carriers are solute-specific transporters. So you're going to have a calcium channel, potassium channel, or a carrier for a couple of different ions, but they're specific. They don't just let any molecule through. If there isn't a transporter, then a water-soluble solute can't cross the membrane. We talked about it being a lipid bilayer. If it's lipid and you're water-soluble and you don't have a channel or a carrier, you're stuck. That solute cannot cross that membrane. Except, there's always exceptions, water. Water it's so tiny. It's such a small molecule that it can sneak through that lipid bilayer. Even though there are some carriers for water, they're called aquaporins, and we'll talk about those later when we talk about the kidney chapter. The other exception is urea. Urea is also small enough to sneak through, and it's very important, again, in the kidney. So we'll get back to that towards the end of the semester. We always draw these channels and carriers like a model. We're not showing you all the atoms that form this protein. Instead, we try and draw it in a way that's more understandable. So on the left, there's the open channel. It's going to create a water-filled pore. It's going to be able to allow things to go from the intracellular fluid here to the extracellular fluid or backwards. And here's that hinge mechanism I showed you for the carrier, where it's either open to the inside or open to the outside, but never both. Okay, and we always show them in models like this, just like two little beans that open and close, because that's going to help us understand it. I've made a list of how do they differ, a channel and a carrier. So a channel, what's crossing through is going to be faster. Given that it's open on both sides, it's just going to be able to flow through. A carrier is going to achieve the same thing, but it's going to be more slowly. The picture there on the carrier here is of revolving doors. You can't go straight through. You have to go through that piece of door, and then it'll revolve in. What else? Well, a channel will allow tens of millions of molecules per second, and the carrier about 1 million molecules per second, 10 times slower. The channel is kind of like a doorway that's open, but in order to open it, you need some sort of gate. You need something that's going to trigger it to open. And what's the trigger? That trigger, that gate that needs to be opened, is going to either be a chemical, it's either going to be a voltage, or it's going to actually be some sort of pressure that you can actually push it and it's going to open. Okay, so we call them chemically gated channel, voltage gated channel, or mechanically gated channel. And the carrier is more like a revolving door. Channels can actually form holes in the membrane. So there's the tube for you to be able to visualize what it does through that membrane. There could be for sodium, potassium, chlorine, and that channel then is going to allow that solute to pass through. And here is the same, this is a superior view of the same thing. Okay, we're just trying to illustrate different ways of illustrating channels. How are we going to illustrate the gate? Well, the gate is really, again, a molecule that shifts position and doesn't allow that passage, but we're going to show it like a little ball with a string. And when the ball and the string is in place there, nothing's going to come through, and when it's not in place, something can come through. Remember, it really is just atoms arranged into molecules that shift in space and close that up or open it up. And they're going to be triggered by chemicals, by voltage, or by pressure. But let's illustrate it like that for better understanding. What opens the gate? I'm saying it again, a chemical, voltage, or something mechanical, something that pushes it open. Now, a carrier-mediated transport is when the molecule is too big for a channel. It's going to use a carrier. Sometimes it's also more than one molecule. So here, for example, this is a picture of the lumen of, let's make it intestine. It could be kidney too, but let's think of the intestine. And in this part of the intestine, you just ate a lot of a granola bar. There's glucose in there. And this is the cell. And over here, there's quite a bit of glucose because it's a cell that's functioning. And the way that we can bring glucose from a low concentration to a high concentration, which is a difficult thing to do because you're essentially bringing it against the concentration gradient, is to use this kind of carrier that allows sodium to attach onto this protein. It's called a sodium glucose transport protein, CGLT protein. And along with that sodium coming in, it's going to allow the glucose to sneak in with it. So let me show you. In the diagram here, we have the open side. You bring the molecule in, it attaches, it closes, and pumps it back in. I'm going to go back and show you the CGLT. We're going to refer to it again a little bit later. For now, know that glucose can be transported along with sodium in one of these carriers, one of these carrier mechanisms. Here we go. So carriers can transport two different types of solutes at the same time. If they're going in the same direction, we're going to call it a symport. If they're actually being transported in opposite directions, we're going to call it an antiport. Symport and antiport. So the glucose example is, in order to bring glucose against its concentration gradient, I do it with the aid of sodium. And that's a symport. They're both going in the same way. An antiport, you'd bring sodium in and, for example, potassium out, which is the sodium-potassium pump that we will refer to later. So solutes are either diffusing through the plasma membrane, or they are mediated transport, which means I'm using some sort of transport protein. And it could be through facilitated diffusion, which means we explained this before, a type of diffusion that does not use ATP, but it does use a protein, or active transport where it uses both the protein and ATP. And there are some extra videos for you to watch on this. The last one I want to talk about is, what if those molecules are slightly large, so potentially they could use a carrier, but let's make them so large that not even a carrier can bring them into the cell. In that case, it's going to be an entire vesicle that's going to be transported into the cell. An example of that is a bacteria. If, for example, a white blood cell wants to englobe a bacteria and remove it from your system because it's attacking it, it's part of your immune system, how is it going to get a big bacteria into the inside of the cell? The bacteria is not going to diffuse. You can't use a carrier. You're going to just have to englobe it all and essentially just make it blast through that lipid bilayer and break it apart. And then it'll assemble back again. So active transport will use some of these carriers, and it will require ATP. I'm going to just glaze through this slide and go on to the next one. I want to show you one, the sodium-potassium pump, which is one of the most important active carriers, meaning it uses ATP, and it's a carrier, and it's an antiport. Why is it an antiport? Because it's going to move two different molecules and in opposite directions. It's going to pump 3 sodium ions out of the cell for every 2 that it brings into the cell. Okay, so it's a pump, it's an active carrier, and it's an antiport. And it's transporting both sodium and potassium. Again, watch a video on this, a separate video on this. If we look at this sodium-potassium pump in steps, look at this. Intracellular fluid in yellow. We have a space to attach 3 sodium ions. And they will attach. We use ATP. ATPase is phosphorylated, which means you utilize that last phosphate. You use it to change the shape of this molecule. It changes shape, and the sodium is released into the inside of the cell, which is great, but we're not done. Now, 2 potassium ions are going to bind to that same protein in the opposite direction and bring that potassium back into the cell. These diagrams are all in your text. And be sure to watch that animation. Thanks for listening. I'm going to cut it there.